Enterococci exhibit intrinsic resistance to a wide variety of antibiotic classes, including most beta-lactams, and have proven adept at the acquisition of resistance to others, either through mutational events or horizontal gene transfer. An example of an antibiotic with which both play a role is linezolid. Resistance to this oxalizidanone occurs as the result of mutations in the 23S RNA, or by horizontal acquisition of the CFR gene, which encodes a methylase that alters the target site. Perhaps the most dramatic occurrence to date has, however, been the emergence of vancomycin-resistant enterococci, or VRE. Although only first detected in 1988, approximately 30 years after the introduction of this glycopeptide antibiotic, it has significantly complicated antibiotic therapy of enterococcal infections. Approximately 60% of Enterococcus faecium isolates are currently resistant to vancomycin. Vancomycin resistance in enterococci is the result of the acquisition of a cassette of genes, VAN-A or VAN-B, resistance operons, and their transposons. These encode at least 7 different enzymes that modify the terminal dealanine-dealanine to dealanine-delactate of the cell wall pentapeptide, resulting in a 1,000-fold reduced affinity for vancomycin and resultant lack of interference with peptidoglycan cross-linking. Methicillin resistance in Staph aureus first occurred in the early 1960s, shortly after the introduction of methicillin into clinical practice, and is widespread both in healthcare facilities and in the community. MRSA accounts for about 19,000 deaths in the U.S. each year. Resistance to this and other beta-lactam antibiotics is the consequence of the presence of a gene cassette called MEK-A that encodes a low-affinity penicillin-binding protein, or PBP2A. VRSA, or vancomycin-resistant Staph aureus, is due to the acquisition of VAN-A or VAN-B gene cassettes identical to those present in VRE, and has thus far been uncommon. It was discovered in a chronic hemodialysis patient in 2002, and in that case the resistance proved to be due to the acquisition of a VAN-A gene cassette from Enterococcus faecalis. Fortunately, only a small number of cases of such isolates have been identified to date. The infrequently encountered vancomycin intermediate Staph aureus, or VISA, first identified in 1996, is thought to result from a thickened cell wall and disrupted diffusion of vancomycin into the bacterium. These alterations reduce the access of vancomycin to the cytoplasmic membrane targets and allow peptidoglycan synthesis to continue unabated. The result is an overall elevated vancomycin MIC, though sometimes still within the accepted range of susceptibility. Intermediate susceptibility to vancomycin appears to be associated with poor outcomes in bacteremic patients and may be homogeneously or heterogeneously expressed. As I already alluded to in the introduction, the majority of the escape pathogens are gram-negatives. Let's now focus on these resistant organisms. Much of the concern about resistance until recent years has focused on gram-negative expression of extended-spectrum beta-lactamases, or ESBLs, that cause resistance to third-generation cephalosporins, but not to carbapenems. ESBLs arose as the consequence of mutations in the widespread plasmid-encoded TEM1 and SHV1 beta-lactamases. ESBL-producing Klebsiella pneumoniae and other Enterobacteriaceae have become widespread in hospitals and are increasingly found as the cause of community-acquired urinary tract infections. While ESBL-producing strains remain problematic, attention is shifting to Klebsiella pneumoniae carbapenemases, or KPCs, which efficiently hydrolyze nearly all beta-lactam antibiotics and are plasmid-mediated. They have appeared throughout the U.S. and have appeared in other countries as well. Since laboratories may have difficulty in identifying the presence of KPCs, their prevalence may be greatly underreported. In addition to Klebsiella pneumoniae, KPCs are now present in other members of the Enterobacteriaceae. The plasmids carrying the KPC gene may also encode other drug-modifying enzymes. KPC-producing organisms are often resistant to trimethoprim sulfamethoxazole, as well as to aminoglycosides and fluoroquinolones. In vitro susceptibility is often demonstrated to fosfomycin, tigecycline, and polymyxins. Fosfomycin, however, is only available in oral form in the U.S., and use of the latter two antibiotics is often associated with the development of resistance to these agents as well. Outside the U.S., another class of carbopenomases, the metalloenzymes, are more commonly encountered. A recent example is the emergence of the New Delhi metallocarbopenomases, or NDM1. Identified in 2010 in a Swedish citizen treated in a New Delhi hospital, metallo-beta-lactamases also have limited treatment options. Acinetobacter baumannii has emerged as an important cause of antibiotic-resistant nosocomial infections. This organism possesses porin proteins that impede the penetration of many antibiotics, as well as at least 2 multi-drug resistance efflux pumps. Acinetobacter chromosomes encode a cephalosporinase, and they generally possess additional acquired beta-lactamases. Isolates may be, quote, pan-resistant, showing initial susceptibility to tigecycline and polymyxin, but this can be lost during therapy with these agents. Pseudomonas aeruginosa has long been identified as an organism commonly expressing multi-drug resistance. Antibiotic classes affected include beta-lactams, aminoglycosides, carbapenems, and fluoroquinolones. Resistance to polymyxins and tigecycline have also been reported. Enterobacter species are among the spice organisms whose chromosomes contain AMP-C that encodes an inducible beta-lactamase, which is predominantly a cephalosporinase, and that is not significantly affected by available beta-lactamase inhibitors. The other members of the spice group are Serratia, Pseudomonas aeruginosa, indole-positive Proteae, which includes Providencia, Morganella, and Proteus, and Siderobacter. Acinetobacter baumannii, which also encodes an inducible AMP-C, has been included additionally in A spice. The major importance of the inducible AMP-C is that, particularly in Enterobacter, small numbers of depressed mutants that constitutively produce the enzyme in large amounts exist at a frequency of approximately 10 to the 7th. These may be selected during treatment with third-generation cephalosporins, and accounts for the admonition against the use of these antibiotics in the treatment of serious Enterobacter infections. ESBLs may also be present in spice organisms, further complicating both laboratory practice and antimicrobial therapy. This brief summary is necessarily oversimplified, since multiple mechanisms of resistance targeting the same class of antibiotics may be present in the same organism, as for example with Enterobacter, showing both de-repressed AMP-C production and an ESBL. Another example seen with Enterobacter is resistance to ertapenem, but not usually to other carbapenems, when a de-repressed AMP-C coexists with porin changes. The latter reduces concentrations of the ertapenem to an extent sufficient to allow significant hydrolysis of the carbapenem. The episode at the NIH described in the previous module is representative of a global crisis caused by the emergence of antibiotic resistance, with a simultaneous dearth of novel antibiotics in the development pipeline. Infections due to antibiotic-resistant pathogens are estimated to annually add approximately 8 million hospital days, and as much as $34 billion in costs in the U.S. This does not account for the costs in lives lost as we run out of effective antibiotics. For some of the patients affected by the NIH episode, we already have reached a post-antibiotic world.